In this example, I'll focus again on FtsZ. This is a bacterial cell division protein, which again needs to form filaments that then form the contractile Z ring needed for cell division. And when we look at the results for the inhibitory fragment measurements, the fragments tiling across FtsZ, here's what the actual data look like. I'm showing you the inhibitory effects along the vertical axis, where larger negative values of inhibition correspond to greater amounts of inhibitory activity. And then along the horizontal axis, we have the position of each fragment along the full protein sequence, from the N terminus to the C terminus. So each point here is a single 30 amino acid fragment of FtsZ. And you can see we perform these experiments multiple times, so we have also measurements of the error associated with a measurement on any given fragment. Okay, so we see that there are these characteristic peaks of inhibitory activity at specific sites in FtsZ, just as we imagined notionally. And how can we understand what these regions actually map to, what these inhibitory effects actually are telling us, right? So if we go back to the structure of FtsZ, we can see that it's made up of these monomers lined up head to tail. And if we take a closer look at the monomer structure, which I've just colored from blue to red from the N to C terminus, we can identify three approximately reciprocal regions that form the interface between adjacent monomers, which I've just called here sites 1, 2, and 3 on one face of each monomer, and 1', 2', and 3' on the other face. These are the sites that come together to form the filament. So if we map these structural features of FtsZ onto our data from the inhibitory fragment assay, we see that actually the strongest inhibitory peak maps to this interaction site 1, needed for filament formation. And similarly, if we look at the other inhibitory peaks, they may actually map out the whole interface for filament formation, which I'm just showing you here in the context of two adjacent monomers in the filament. So we actually see that these fragments are mapping out reciprocal sites from either side of the interface needed to form the filament. And that's exactly what you expect if these fragments are actually able to inhibit filament polymerization in cells.